Good morning, students. So, this is the second part of your chapter 8, that is human health and diseases. So, first chapter we had seen about the various diseases, its diagnosis, yes, and preventive methods and all. So, I hope you have gone through that part. Now, in this part, we will be seeing about a little about immunology, so immunity. Okay. So, immunity basically, if we define immunity, it is the ability of a body to defend itself from different types of pathogens or foreign bodies. Okay, that is immunity. So, every body has a defense mechanism, so that is called immunity, and the study of this process is called immunology. So, ability of the study of the ability of an organism of a person to resist against diseases, that is immunology. So, that means immunity is also called as the body's resistance to diseases. Alright, now coming to different types of immunity. There are broadly, if you see, there are two types of immunity: innate and acquired. So, innate is the natural immunity that is present in a person from birth, since the time of birth. Okay, so it is naturally obtained, whereas acquired is the immunity that is acquired or present in a person in the lifetime. During the lifetime, he receives that, or he acquires the immunity, that is called acquired immunity. So, in innate immunity, as I told, it is natural or non-specific. It is not specific to a particular disease or a particular pathogen. It is non-specific and it is inborn immunity. Okay, so in that there are four barriers are present. Four barriers that provide innate immunity. One is physical barrier, then physiological barrier, cellular barriers, and cytokine barriers. Okay, so in physical barrier we have different parts of the body like skin, then glands like sweat and sebaceous glands, mucous membrane, nasal hair, cilia. So all these provide first line of body's defense. Okay, that means physical barriers. So skin, we know it is a protective layer or a barrier for the entry of any kind of germs. Sweat and sebaceous gland, the secretions, they secrete substances which are bactericidal or fungicidal and kill the bacteria or fungus. Similarly, mucous membrane also, they secrete mucus, or mucus is secreted by the glands, which is present in buccal cavities, nasal cavities, alimentary canal. So, all the tracts, it has mucus which traps the pathogens and prevents the entry of these pathogens into the internal organs. Then we have nasal hair. So, the nasal hair, it traps the dust and the pathogens that are present inside the dust from entering into the respiratory tract. Then we have cilia. Cilia is present throughout the windpipe to trap the bacteria. So, in the windpipe, I told there are mucus is also present. So, along with the mucus and the continuous movement of cilia, it sweeps away all the pathogens. Okay. Now we have physiological barriers. So these basically contain some secretions or the productions. So they have the lysozymes which are produced by saliva, mucus and tears. They contain bactericidal they have bactericidal effects which break down the bacterial cells. Then cerumen or earwax. So earwax, it's a sticky substance produced in the ear which prevents any kind of pathogens from entering into the internal layers of the ear. Okay, so it traps these pathogens. Then we have acidity of gastric juice. Acidity of gastric juice is basically the acid that is produced in the stomach. Okay, that acidic condition, it kills the microorganisms that reach the stomach. Then we have alkalinity of bile juice. So alkaline nature of the bile, it prevents the growth of any microorganisms in the intestine, mainly duodenum. Then there are other secretions like vaginal secretions which prevent the entry of or they kill the bacteria. Then the body temperature which regulates the helps in the production of different toxins to prevent the entry of pathogens. Then there is sweat and oil produced by the glands that I already discussed. Okay, so now coming to the cellular barriers. Cellular barriers, there are a few cells. So these basically they are the cells which are present in the body which act against the pathogens or microbes. Okay, so first we have leukocytes. Leukocytes is nothing but the white blood cells. So we have already studied about white blood cells. They have this defensive mechanism. Yes. So in leukocytes, mainly neutrophils and monocytes, they act as or act against the microbes. Alright, so neutrophils, they are also known as polymorphonuclear leukocytes. So they, whenever there is any microbes that enter into the body, in that area, the neutrophils squeezes out of the capillaries. The squeezing out process is called diapedesis. So by diapedesis, they reach the site of infection and they engulf the bacteria or microbes. Okay, so these are the first cells that arrive the site of infection. Next is monocytes. They are large amoeboid leukocytes which have phagocytic property. So what is meant by phagocytosis? Phagocytosis means they engulf the microorganisms and any foreign particles. Yes. So after a certain time, so after maturing, after entering into tissues, these monocytes change into macrophages. Okay, so these are other next cells which are present in the cellular barriers. So macrophages, there are two types: mast cells or fixed macrophages. Mast cells, also known as wandering macrophages, name itself it is clear, they keep moving, they are in continuous movement in the connective tissue throughout the body to reach the site of infection. Okay, whereas fixed macrophages are the macrophages that are located at a specific location like lymphatic capillaries, spleen, liver, bone marrow, all that. Alright, so these are about macrophages. Now coming to natural killer cells. Natural killer cells are the cells, cytotoxic lymphocytes, which kill the infected cells, which are already the cells which are already infected with virus or tumor. They kill these cells. Okay. So what happens whenever there is an infection in a cell? These NK cells target the cells. So these cells are called target cells, and they produce a substance called perforins. So what is the function of perforins? These perforins create pores inside the target cells. So inside the target cell, when there is pores, water enters and the target cell swells up. So after gradual swelling up, it bursts out. So that means in this way it kills it destroys the infected cells. Okay, so it prevents the other cells from being damaged. So cellular the remains that are present after the bursting, these are eaten by the phagocytes. Okay, by phagocytosis. So here you have to know the process of phagocytosis, how it is taking place. Next is cytokine barriers, or it is also known as interferons. So these are specifically they are species specific, and they are work specifically against viral infections. Okay, the cells that are infected by virus, they produce interferons. They are a kind of antiviral protein which is produced by the cells infected by virus. So that is a natural mechanism that is present in the cell. Okay, so these interferons, when they are released from the infected cells, these interferons prevent the other adjacent cells from getting the infection, the secondary, that means from getting this viral infection being transferred or transmitted from this cell to other adjacent cells. These interferons prevent by inhibiting the multiplication of viral particles. Okay, so how this mechanism, how interferons work, they inhibit viral replication. Okay, they activate the NK cells and macrophages. Yes, so that means what is the function of NK cells? To kill or to destroy the cells. So those interferons, after they are produced from the infected cells, they activate the NK cells so that they can destroy these particular cells. Okay, and macrophages, because they reach the site of infection, and phagocytosis takes place. This also increases antigen presentation to lymphocytes. Okay, so there are some cells of the immune system which present these antigens to the lymphocytes. Lymphocytes act on these immune cells or the antigens. So these immune cells, that is lymphocytes, they have to, in order to act on these antigens, they are presented by the antigen presenting cells. So whenever interferons are produced, it increases the antigen presentation. So recognition, it helps in the recognition of these infected cells by the T and B lymphocytes. Then it induces resistance of the host cells to viral infection. Okay, it increases the resistance against virus. Okay, so since interferons, I already told they are species specific. Okay, so since interferons is a useful protein or substance that is produced, they are artificially produced by rDNA technology. So we have already learnt about recombinant DNA technology. So they are produced by rDNA technology and helped in the treatment of various viral diseases like hepatitis and influenza, and even in some cases against cancer also. Okay, so that was about interferons. This innate immunity, we have already seen the different barriers that are present in innate immunity. Okay, so now we'll move on to the acquired immunity. Acquired immunity, I told that this is the resistance against a disease that a person acquires during his lifetime. Okay, so here also they are pathogen specific. Okay, innate, I told they are non-specific, they are not specific. But here acquired immunity, this is specific. So it is pathogen specific, and it is also known as specific immunity or adaptive immunity. It yes, so it comprises of antibodies and lymphocytes. So the production of antibodies against the antigens and the action of lymphocytes. Okay, so we will be seeing about all this in the acquired immunity. So what are the features of acquired immunity? First is specificity. So I told it is specific to each type of pathogen. Diverse property it has, that means the acquired immunity can develop against diverse types of pathogen. Okay, Now, discrimination between self and non-self. So I told acquired immunity is immunity developed in the course of lifetime, or in the during the lifetime it is acquired. Okay, so in that time the self cells and the non-self cells, that is the self, that means the body cells, body's own cells, and the non-self, that is foreign particles, are differentiated. Okay, so if this property fails to function in the immune system, that leads to a disorder or dysfunction called autoimmunity. Autoimmunity means auto self. So the immune system of the body starts acting against its own body cells. Okay, so it is not able to discriminate or differentiate between self and non-self cells. Okay, next we have the memory. So immune system of a body has a specific feature that it can recognize a foreign antigen, a foreign agent, foreign substance after its first encounter. So first time when it has entered into a body, the immune system will act against it and remove it. If the same antigen enters the same body again, the immune system will recognize. So the antibodies or the substances that were produced in the previous invasion or previous encounter, that same antibodies will act on the during the second encounter. Okay, so that is what is meant by memory. Okay, now coming to the components of acquired immune system. So components of acquired immune system, there are two components. One is antibody mediated immune system, or which is also known as humoral immunity, and the other is cell mediated immune system. So we will be seeing about antibody mediated in this part. So antibody mediated immune system, that is basically the production of antibodies. Okay, it consists of different types of antibodies. So antibodies occur in the body of plasma, which are formed in the plasma cells, and these plasma cells are produced by the B lymphocytes. Okay, so when B lymphocytes are activated during any kind of infection, that time B lymphocytes now triggers the production of antibodies. Okay, so produced antibodies are specific against specific kind of antigens. All right, so what are the roles, what is the role of humoral immunity? They defend the body against toxins entering the body, bacteria which have polysaccharide capsule, that means protective layer, and also against viruses. Okay, so this antibody mediated, that means by the production of antibody the immune system works. Next is cell mediated immune system. So here it consists of T lymphocytes. So in antibody mediated, I told it is B lymphocytes are involved. Here T lymphocytes are involved. Okay, so these T lymphocytes recognize the infected area or the infected cell. Yes, then they activate the B lymphocytes. So when B lymphocytes are activated, what will happen? B lymphocytes will trigger or activate the production of antibodies in the plasma. Yes, so they are this both humoral immunity and cell mediated work side by side, but they are different components. It's humoral deals with the antibody production, and here cell mediated, that means cells are at cells work in the process, cellular immunity, okay, against the pathogens. So it provides cellular immunity. Okay, it helps in the destruction of cancer cells. Okay, So it, as I told, it also triggers or activates the B lymphocytes. Okay, so difference between innate and acquired immunity you have to know, and also cell mediated and antibody mediated immune system. So this small differences you will have to know. Okay, now coming to antigens and antibodies. So first we'll see what is antigens. So antigens means these are the foreign substances which on entering the host cell stimulates the immune response. Okay, so that means whenever a foreign substance, that means antigen, enters the body, it triggers the body to immune response is produced, in which this immune response leads to the formation of antibodies, which is specific to this particular antigen. Okay, so the ability of an antigen to stimulate the antibody production is called antigenicity. Clear? So what is antigenicity? These certain terms you have to remember. What is antigenicity? Ability of an antigen to trigger or stimulate the formation of antibody. Alright, next is complete antigens. What are complete antigens? So antigens, the composition you have to know. Antigens can be proteinaceous, made of or can be made of carbohydrates, lipids or nucleic acids. Okay, so the proteinaceous antigens, so antigens which are proteinaceous are called complete antigens because they can induce antibody formation. Okay, in other words, the antigens which can induce antibody formation are called complete antigens. Yes. So what are haptens? So haptens are the antigens which are not able to trigger the production of antibodies. Okay, those are called haptens. So that means haptens, if you see the composition, haptens are made of lipids and carbohydrates. Okay, they do not have proteins. So these these antigens which contain lipids or carbohydrates and are unable to produce or trigger the production of antibodies are called haptens. Okay, but there is a case where they can produce when hapten is combined with a protein. So hapten when combined with a protein can lead to antibody formation. Okay, so here you will come across two terms, that is epitope and paratope. So epitope is the site present on the surface of the antigen which is recognized by the T or B cells or the antibodies. Okay, the site where the antibody attaches to the antigen. Okay, site where of recognition present on antigen. Okay, epitope is present on antigen, and on the same way paratope is the site present on antibody which has receptors for which serves as the receptor for epitope. Okay, so these two terms you have to know, epitope and paratope. Alright, so now coming to antibody. So antibody, they are also known as immunoglobulins. Okay, they are formed for self defense against antigens. Okay, so whenever I told whenever there is any antigen attack or and pathogens enter the body, the self defense, that is the production of antibody. Okay, so if you see the structure of antibody, it is Y shaped and it has 4 peptide chains. Okay, 4 peptide chains. This is 1, this is 2, 3 and 4. Okay, so 4 peptide chains, out of which 2 are light chains and 2 are heavy chains. Okay, so the heavy chains, they are called heavy chains because they extend to the stem. If you can see the stem of Y cell, the Y shaped antibody, the stem has heavy chains, both the heavy chains, but light chain is present only on the arms. Okay, and here you can see the antigen binding site. Okay, so this is, so it is specific. I told it is antigen specific, so each antigen binding site and will be different. All right. So these all the chains are joined together by disulfide bonds. So how are they? How are the light and heavy chains joined? They are joined by disulfide bonds. So you can see here they have disulfide bonds. Yes. Okay. This is called the V region. Okay, sorry. This is called the V region. Okay, or the antigen binding filament. So this filament helps in the antigen binding, and this is the FC or crystalline fragment. Okay, so that is the structure of basic structure of antibody. Now coming to the classification of antibody, there are basically 5 types: IgA, IgD, IgE, G and M. All right. So IgA, out of all the antibodies, 10 to 13% is IgA. Okay, it is called secretory immunoglobulins because it is present in all the body secretions: tears, urine, saliva, seminal fluid, mother's secretion, colostrum. So colostrum is the first milk or first the substance that is produced after delivery for the child. Okay, so that also gives immunity to the child. So that is secretory immunoglobulin. It provides first line of defense. So if you remember, in innate immunity I had told these secretions, yes, physiological barriers, that is the secretions, provide first line of defense. Okay, it is effective against a number of antigens, especially ones with polysaccharides. All right, so IgD is just 1%. It is effective against toxins and allergens, and it activates the B lymphocytes. So once B lymphocytes are activated, the B lymphocytes help in the secretion of other immunoglobulins. So indirectly IgD is necessary for the production of other immunoglobulins also, because it triggers or it activates the B lymphocytes. Okay, now coming to IgE, it forms only 0.002%, but it is very important because it triggers hypersensitive reactions. Okay, so how this happens? By inducing basophils and mast cells to release histamine. So mast cells, we have learned about mast cells, that is macrophages, type of macrophage. It releases histamine whenever there is a hypersensitive reaction. So histamine, when it is released, it produces inflammatory or any kind of reactions. Okay, that is IgE. Now coming to IgG, so this is 75 to 80% of the antibodies is IgG, and it gives a long term resistance. So this is the only immunoglobulin which can cross the placenta. Okay, so it provides immunity to the fetus, right? So IgG of the maternal antibodies can cross the placenta and provide passive immunity to fetus. So we will be discussing about active and passive immunity in the next video. Okay, so this IgG is the, we have to remember the main characteristics of all these immunoglobulins. Okay, so like IgG, it can cross the placenta. So which is the immunoglobulin that can cross placenta, which is the immunoglobulin present in colostrum, which activates B lymphocytes. So all those you have to know. Okay, these are